Good afternoon. My name is Deborah Johnson, and I am a branch chief in the Office of New Drug Product in the Division of Lifecycle API. And today I will be discussing nitrosamines and where we are now, what we've learned. So I will discuss the lessons learned, and I will provide suggestions for future considerations, and I will briefly discuss the agency's nitrosamine guidance. In early 2018, a tiny peak was detected in a residual solvent test in a GC chromatogram. Because the peak was showing up consistently, the firm decided to identify it, and they eventually discovered it was N-nitrosodimethylamine, which the shorthand for that is NDMA. The detection of that tiny peak has resulted in an investigation which has been ongoing for 24 months. It started out as just valsartan. It extended to all of the sartans, and now it's expanded to include multiple APIs and drug products. So after a couple of years of investigations by firms, countless inspections, hundreds of information requests and complete response letters, what have we learned? One thing we have learned is that process understanding should extend to all reaction components and their interactions. That means thinking holistically about the fate and potential interactions between starting materials, reagents, solvents, and catalysts. Sometimes we assume that residual reagents, solvents, and catalysts are removed during workups between steps. But what we have learned is that these kind of reagents and impurities may carry forward farther than we had anticipated. It is also important to understand how all of the components could potentially interact. For example, NDMA and NDEA, which is N-nitrosodiethylamine, can be generated inside reactions between sodium nitrite or other nitrosating agents and dimethylamine, which might be present as a breakdown product of dimethylformamide, or triethylamine, which is a commonly used organic base. These side reactions are completely unrelated to the synthesis of the API, and they're happening in the background. Something else that we've had to start answering is how do we show comparability of pre- and post-change material? First, we shouldn't assume that meeting the original specification is sufficient to show that pre- and post-change materials have equivalent quality, although this seems to be the preferred method in the submissions that we see. The specification was designed for the process as it was originally developed. Once the process has changed, a reevaluation should be done to consider what new impurities might have formed that were not controlled in the original specification or were not present as part of the impurity profile. It is important to remember that new impurities may not be detected by the current analytical methods due to such issues as a lack of a chromophore or poor volatility. If there are new potential impurities that can be formed, then some new analytical methods may need to be developed. Those methods may or may not be eventually become part of the specification. In the draft post-approval changes guidance for drug substance, which was sent out for comment, is in back with the agency, we discuss this idea of comparability between pre- and post-change material. One thing that we have discovered is that the post-change impurity profile should be compared to see if there are differences. So watch the number and the RRTs and the levels of unknown impurities. Can your process now produce new PGIs? And if so, can your current methods detect them? After change, if you're seeing small unknown peaks, which are showing up consistently, it might be a good idea to take a look and understand what those are. Because as we've discovered, small unknown peaks can hide big problems. So if your process there's a potential for some kind of PJ to form, it's probably a good idea to consider whether that's something that you're seeing with these small unknown peaks. So this is just an example of a reaction scheme. And in this drug substance, they discovered that it was contaminated with NDMA. So you might ask, well, how did that happen? Well, if you look at the synthetic scheme, you'll notice in the first step you're using sodium nitrite. And in the very last step, you're using dimethylformamide. So the theory is that sodium nitrite, though there were water washes and it should have been removed, somehow some residual amounts of that material carried through into the step where there was dimethylformamide. There was probably some dimethylamine that was present as a breakdown product, and there were acidic conditions. Because you now have all the components necessary for a nitrosamine to form, NDMA did indeed form. It was actually at levels that was detectable. So this is just an example of how we need to understand that, if there are precursors in the process, even though they're separated by some steps, one cannot assume that there is no risk. So something else that we've discussed, or that we have discovered, is that process understanding needs to extend to recovered solvents. This is especially important after a process change. ICH Q3C does not have a test, or does not have a limit for any unknown impurity. That's different than ICH Q3A and ICH Q3B. So there's no limit for this. So if you've done a process change and you're seeing a new peak, we need to ask the question, is this a peak that belongs to an impurity that is controlled under another test? Perhaps it's something that's controlled through the related substances HPLC test, or maybe it's controlled through some kind of other GC or mass spec test. And if it is, then this peak is not a problem. We also need to understand which peaks belong to process solvents and byproducts. So if you're using a process where you're using methanol in one step and ethyl acetate in another step, it's possible that you could form methyl acetate. So if the peak belongs to something like a byproduct of two solvents, then it's probably not a problem. We also need to understand which peaks belong to the mobile phase and the diluent. It's important to use solvents of appropriate grade, because if those solvents contain a large number of peaks, especially if the peaks are of considerable size, they could be masking impurities that are coming into the process through the API. Another thing is we can't assume fresh solvents are clean, which seems counterintuitive. However, when you're choosing a new vendor, you need to exercise due diligence. Understand what is happening at that plant. Some of those vendors are creating solvents from multiple industries, and some of those products might be fairly toxic. Another thing to consider is the vendor bringing in used solvents and recycling them. How are their tankers cleaned in between? That's especially important because, as I mentioned, some of those other materials can be quite toxic. So these are just some questions that you want to consider when you're vetting a new vendor. So this is just an example. So if you'll notice at the top, there is a blank chromatogram, and notice that the baseline is nice and flat. Directly below is the chromatogram for specificity, where the process solvents have been spiked at their limits. And again, the baseline is nice and flat. All of these chromatograms have roughly the same scale. But on the right, this is precision. So this is now where the API has been introduced into the sample, and if you'll notice between acetone and dichloromethane around 8 minutes, there is a new peak. And that peak is approximately the same size, or maybe even slightly larger than that of dichloromethane. So it's not a peak that is insignificant. So is that peak important? Well, it depends. If there's been a process change and this is a new peak, it might be important. Does it belong to an impurity that's controlled through another test? Is it a byproduct of a reaction between two other solvents? If there's a potential PGI and that PGI is not detected by any other test, then this could potentially be a problem. So especially after process change, if you're picking up peaks in your recovered solvent test and they're consistently, and they're in this case they're approximately the same size as one of the solvents you're controlling, it might be a good idea to go ahead and investigate and see whether that is a peak that is already controlled, or is this something new that needs to be controlled in some other way. So some other things that we've learned, that we understand that nitrosamines are formed when there are the precursors, so amines and a nitrosating agent. But sometimes those precursors come into our system in ways that are not obvious. They can be hidden. For example, if you're using sodium azide in your process, sodium azide contains a considerable amount of sodium nitrite. It can be hundreds of ppms, and it can vary widely between vendors. So if you're using sodium azide and there are amines present either in the same step or later steps, you then now have the precursors for nitrosamine in your process, and you cannot assume that, these aren't forming. So in this case, you probably need to assume that they potentially are forming and do some work to see if their nitrosamines are actually there. Another potential place for amine precursors to come into your system is as contaminating amines in amine bases or catalysts. For example, triethylamine can contain a fairly high amount of diisopropylethylamine or isopropylamine or isopropylethylamine. So there's two secondary amines that are coming into the process that are not potentially expected. Or if you're using tetrabutyl ammonium bromide, that can contain dibutylamine or tributylamine. So need to be aware of where potentially other amines are coming into your process. Another place where you might be getting a secondary amine is through degradation of amide solvents. This is especially important if the amide solvent's being used at high temperatures for extended amounts of time, or if it's in the presence of water, acids, or bases. You could be generating a fair amount of a secondary amine. Amine contaminants can also be present as impurities as coming in from purchased starting materials or intermediates. So it's important to understand what kind of amine contaminants or nitrite or other nitrosating agent contaminants can be coming in through purchased materials. Secondary and tertiary amine functional groups might be on your starting material, your intermediates, or your API molecules. And if there is the potential for a nitrosating agent product in your process, then you have the potential to form a nitrosamine. So again, it's thinking holistically about all of the materials that are coming in. What do you know is there? What things might be there that are hidden? And in this way, you can prevent a surprise at the end of the process. So I just want to use this synthetic scheme as an example. This is varenicline tartrate. It's taken from a public source, so there's nothing proprietary here. And the question is this synthetic route at risk for nitrosamine formation? There's a lot going on in this process. So take a look and see if you can tell whether or not this process might be at risk for nitrosamine formation. Well, since I'm asking the question, the answer is probably yes. Yes, it is at risk. And so where is the potential for nitrosamine formation coming in? Well, if you look at the first step, there was a key starting material that has a secondary amine. And the first step of the reaction is where that amine is protected by trifluoroacetyl group. But if some of that starting material actually carries forward and it's not protected, then it can go into the next step where you're using nitric acid to put the nitro groups on the benzene ring. And nitric acid can contain, a fair amount of nitrous acid. So if that secondary amine contacts nitrous acid, you can form a nitrosamine, which is compound 3A. Now, compound 3A looks a lot like its derivative that's supposed to have a trifluoroacetyl group on that amine group. But the part now that's reacting are the nitro groups, which are supposed to be reduced by hydrogen and palladium to amino groups. Well, this can go straight through and form a nitrosyl derivative of that intermediate, which is 4A. And then the amine groups react with glycol to form the varenicline, but since those amino groups have the same reactivity as the actual intermediate, it can go straight through the reaction and form a nitrosyl version of varenicline. So it is actually possible that you could pick up 3 different nitrosamines in this process. But they weren't obvious. This is because an impurity, that residual key starting material, carried through into a step that had nitric acid. So this is why it's important to think holistically about the process. You do have precursors in the process. If there's precursors in the process, even if they're separated by a step or two, you cannot assume that nitrosamines aren't forming. So another lesson that we learned were faulty assumptions. So sometimes when we're looking at a process, we may assume some things, and we have seen some missteps in that, in assessing risk or process capability. So as I mentioned earlier, mostly nitrosamines are forming as byproducts. So these are things that are forming in the background. When things are forming in the background and they're not part of the normal synthetic process, they're forming in different amounts from batch to batch. There's no real consistency in how much might be forming. So that makes doing a spike purge study or assessing process capability difficult. Normally, when we do these things, we start from the knowledge of how much is potentially forming. And based on that knowledge, we can do a spike purge, or we can do some theoretical calculations to determine the process purging capability. But if we don't know how much forms from batch to batch, it's going to make those assumptions or those studies difficult. Something else that we've discovered, the purging capacity of these nitrosamines appears to be much lower than you would predict. For example, NDMA is water miscible, and it's soluble in organic solvents. Well, since most processes contain multiple aqueous washes of organic phases, one might assume that something that's water soluble would be completely removed from the process. However, it's not that easy. There's been some studies done that actually show that the NDMA prefers the organic phase, and it's not being purged as effectively as one might think through aqueous water washes. So if one is assuming that the impurity is completely miscible in water, that could create a possibility that a theoretical purge calculation is overestimating the purging factor of that process, and instead of being totally removed as one might think, it could actually carry through. Another instance is the reactivity of nitrosamines might be overestimated. So in the varenicline tartrate synthetic scheme, after the nitrosamine was formed, there was a reduction with palladium and hydrogen. One might assume that nitrosamines reduced to the hydrazine. However, nitrosamines may not have the same level of reactivity as one assumes. So the idea here is you can't assume. You may need to do some tests. Make sure that the solubilities are what you think. Make sure the reactivity is what you think. So just don't assume. I'd like to just discuss very briefly now the nitrosamine guidance. It was published on September 1st, 2020. And in that guidance, it contains information on the acceptable intakes for 6 nitrosamines. But we all know there are more than 6 nitrosamines out there. So if you have a process that creates a nitrosamine that is not listed in the guidance, then you need to follow ICH M7 to produce the information that you need to support the proposed control strategy for your nitrosamine. Please remember that the threshold of toxicological concern does not apply to nitrosamines because they are part of the cohort of concern. These are much stronger genotoxic impurities than normal PGIs. So you cannot just assume that TTC is the relevant acceptable intake. So you'll have to follow ICH-M7 to develop the information you need to support a potential level or limit. The other thing that is discussed in the nitrosamine guidance is that it establishes a limit for total nitrosamine at 26.5 nanograms per day based on the MDD of the drug product. This is applicable when the number of nitrosamines in the specification is greater than 1. Because it's not well understood how nitrosamines interact, if your process can produce more than 1 nitrosamine and they're controlled in the specification, and there is a potential for the API to contain more than 1 nitrosamine, then out of an abundance of caution, you need to add a test for total nitrosamine, and that limit needs to be set using the acceptable intake of 26.5 nanograms per day. Also, your control strategy should focus on what nitrosamines can be formed in your process. We all know that there's a lot of nitrosamines that are known out there now, but not all of them are actually going to be formed in your process. So your control strategy should be focused on the nitrosamines that are possible. But remember that part of that control strategy is considering things like your fresh solvents, making sure that those are of appropriate quality so you don't get a nasty surprise at the end. With a process that's supposedly clean and risk-free, having nitrosamines only to find they came in through fresh solvents or because they came in through a purchased material. So again, thinking holistically about all the things that can happen in the process and all the things that could be coming into the process from the outside. Another thing that we discuss in the guidance is the analytical methods. Because nitrosamines are typically controlled at much lower levels than other kinds of impurities, we recommend that your limit of quantitation be not more than 0.03 ppm. However, if you have a high dose drug product or you have multiple nitrosamines present, then you may need a limit of quantitation that is lower than that in order to be able to accurately quantitate the levels that you have. So in the guidance, the first thing that happens is you do a risk assessment. And if in your risk assessment you discover that nitrosamines are theoretically possible, the next thing that's supposed to happen is confirmatory testing. So if you've done your testing and you are seeing a peak that might be that nitrosamine, it's below the LOD. So it's there, but it's below the LOD. You should probably do a root cause, because even if impurities are forming at very low levels, you need to understand where are they forming, how much are they forming, where, and how well does your process purge them, because you need to make sure that your process can consistently reduce these to a level in the API that's not concerning. Now, if you do the confirmatory testing and you actually see nitrosamines that are above the limit of quantitation but below the acceptable limit, then routine release testing is recommended. So which control strategies might you use? Well, ICH-M7 has options 1 through 4, and you are welcome to use all of them. You are welcome to add a test to the specification, which is option 1, but you may also use options 2 through 4. However, those need to be supported by sufficient data. And the amount of data that you need to support your impurity control strategy is going to go up with the increasing proximity of the impurity to the final API. I will tell you, though, that when impurities are formed in or introduced at the final step, that a completely theoretical purge calculation is not sufficient. When these types of impurities are added in or formed in the final step, then you will need actual testing data to support any kind of proposed control strategy. And that is all that I've got. I'd like to acknowledge my division director, David Skanke. I'm also a member of 2 different nitrosamine task force. We've worked together for 2 years. A lot of our knowledge has come through discussions through them, so I'd just like to acknowledge them. I'd also like to refer you to another presentation, the Safety Evaluation of a Drug Substance Impurities in Generics by Chanchal Gupta. Also, please remember to type any of your questions that you have into that Q&A box so that we can answer those in the live Q&A session following this. And if you have a question that wasn't answered or we didn't get to it, and you'd like to have us take a look at it, please submit those questions for inclusion in our follow-on webinar on April 9th. Please send them by March 19th to dmfworkshop2021 at fda.hhs.gov. Again, thank you. I appreciate your attention.